Good day, ladies and gentlemen. My name is Sedzani Martin. I am a master's student at the University of KwaZulu-Natal. So today I'll be sharing with you my research project titled Investigating the Gut Dysbiosis during Uropathogenic Escherichia coli-Induced Urinary Tract Infection and Microbiome Diversity during treatment with Manihot esculenta. So I'll just start off by introducing the gut microbiome. So the gut microbiome or gut microbiota is basically a collection of microorganisms such as bacteria and archaea. These microorganisms inhabit your digestive tract and are very diverse, being made up of hundreds of different species. So the bacteria within the microbiome contribute to the breaking down of complex carbohydrates, fibers, and other indigestible compounds. And this breaking down results in the production of short-chain fatty acids as well as other metabolites that serve as a crucial source of energy for the body. The gut microbiota also has an influence in the metabolism of nutrients, including the absorption of essential vitamins and minerals. It also plays a role in regulating energy balance and may impact things like weight and where fat is stored in the body, fat storage. So the gut microbiota also interacts with the immune system, influencing its development and function. And beneficial bacteria help to maintain a balanced and responsive immune system, helping to protect against infections. So bacteria in the gut microbiome also contribute to your intestinal integrity, helping boost the intestinal barrier by preventing harmful pathogens and toxins from coming into the bloodstream. So the bacteria, although carbohydrates are converted into short-chain fatty acids, undigested carbohydrates will be fermented. There will be bacteria that will ferment your undigested carbohydrates into gases like hydrogen and methane, and this process helps to influence your gastrointestinal motility. So on this slide, we can see a couple of groups of bacteria that are dominant in different regions of the body. So in the colon or large intestines, we can see that we have bacteria such as Enterobacteria, we have Enterococcus, we have Fecalis, we have bacteria such as bacteroids. So these are very crucial in managing inflammation in the body. We have bacteria such as Peptococcus, we have Ruminococcus, Clostridia, and Lactobacilli. So all these bacteria work together to maintain homeostasis in your gut microbiome. So in summary, the gut microbiome is a dynamic and complex community of organisms, and it serves to contribute to your good health. Maintaining a balanced and diverse gut microbiome is essential for your overall health and ability to prevent disease. So now we come to urinary tract infections or UTIs. These are the most common type of infection in humans with a prevalence rate of approximately 90%, affecting approximately 60% of females and only 40% of males. So UTIs can be caused by a variety of microorganisms such as Escherichia coli, Staphylococcus cytophyticus, Klebsiella pneumoniae, Enterococcus fecalis, and Proteus mirabilis. So E. coli, amongst the lot, E. coli is the most abundant type of microorganism in the human gastrointestinal tract, and it usually forms a symbiotic relationship or a good relationship with the gut. And non-pathogenic Escherichia coli is harmless bacteria and rarely causes any complications in the body. However, in cases where the host is immunocompromised, this will result in an infection, typically a uropathogenic Escherichia coli tract infection, which is a common and often painful bacterial infection of the urinary system, primarily caused by the E. coli strain. Common symptoms of UTI infection include, but not limited to, burning or painful urination, constant urge to urinate, cloudy urine, found foul odor, pelvic pain in women, amongst others. With urinary tract infection having such a high prevalence rate, there have been a multitude of treatments that have been established. Most common are antibiotics such as norfloxacin, amoxicillin, cephalexin, and ciprofloxacin, just to name a few. Ciprofloxacin was among the first to be highly or readily accessible, and it was widely successful in treating a variety of bacterial infections, ranging from UTI, sexually transmitted infections, all the way to gastrointestinal infections. However, it does have side effects such as hyperglycemia and hypoglycemia, and also photosensitivity. In the never-ending battle to try and control the prevalence of UTIs, alternative methods of treatment have been explored. Treatments such as cranberries, vitamin C, odyssey, Manihot esculenta. Manihot esculenta, also called cassava, is a good source of resistant starch, and it supports gut health and blood sugar management. Based on some research, resistant starch or slowly absorbed starch have great impact on people's health. The colon is where the microorganisms digest the resistant starch components and thus result in the production of short-chain fatty acids, which help provide the body with energy. Because of this, treatments that involve or include resistant starch have beneficial effects on patients with diabetes, certain cancers, cardiovascular conditions, and intestinal health. Cassava or Manihot esculenta also contains vitamin C, and this is a key micronutrient that can enhance the immune function and collagen production. So now we come to the methods and materials section. So ethics was acquired from the Animal Research Ethics Committee, and that's the AREC number. The aim of the study was to evaluate the effect of UPEC-induced UTI on the gut dysbiosis and microbiome diversity treatment during treatment with Manihot esculenta. So we had a total of 35 rats divided into a group of 5. So the first group was the benchmark group or the control group, which had no infection and no treatment. The second group had an infection but no treatment. The third group had an infection and was treated with our antibiotic or benchmark antibiotic, which is ciprofloxacin in this case. And then the fourth group was infected and then treated with our cassava or Manihot esculenta. And then the final group was infected and treated with a combination of our antibiotic as well as our medicinal plant. So after infection and treatment, the rats were euthanized and the fecal matter from the large intestine or colon was collected. Specifically, the cecum part of the large intestine was collected. And total DNA extraction was performed using the Zymobiomics DNA mini prep kit, and the samples were sent off for metagenomic sequencing. The results were then loaded and analyzed on KBase following the KBase protocol. So after running through KBase, we were able to plot the graph below. So the first bars represent our uninfected and untreated group. So that is our benchmark group. And then we can see that during infection, there was an increase in oscillospirals and bacteriodale groups of bacteria, and a decrease in Lachinospiral and Lactobacillus. And for ciprofloxacin, Manihot esculenta, and the combination treatments, we had the same trend, just in different intensities. We can see that in all of the three, we had an increase in bacteriodales, Lachinospirals, and Lactobacillus, and a decrease in oscillospirals. So we had 4 different groups of bacteria that were mainly involved. So we had oscillospiral, which are linked to constipation, which is a symptom of an infection. So there were no traces of bacillospiral in the original, in the untreated and uninfected group in our control group. However, they shot up to their highest level when the infection was introduced to the body, to the host. When the infection was introduced to the host, we saw that the levels elevated all the way up to 19%. However, the three medications or the three treatments were able to lower the levels of bacillospiral to 8, 8, and 13 respectively, meaning that the host will be less likely to suffer from constipation. And then the next one in line is our bacteroidal. Bacteroidal is responsible for converting carbohydrates into short-chain fatty acids, which provide the body with energy. And that energy will be used to fight off inflammation. Low levels of bacteroidals are usually associated with gastrointestinal disorders such as inflammatory bowel syndrome. However, we can see that the three medications were able to elevate the levels of bacteroidals in the body, meaning that they gave the host a better chance at fighting off any potential inflammation. And then the next one is our lichenospiral. Lichenospiral is an anti-inflammatory bacteria or group of bacteria. So we saw that when the infection or the UPEC strain was introduced, we saw lichenospiral levels drop from 33% to 21%, meaning that the host is less capable of fighting off an inflammation than they were before their infection. However, ciprofloxacin was able to elevate the levels up to 31%, and Manihot esculenta was able to elevate the levels up to 21, from 21 to 24, and then M. esculenta and ciprofloxacin combination raised the levels all the way up to 42%, giving the host a better chance at fighting off any potential inflammation in the body. And then the last bacteria is our lactobacillus bacteria. So this bacteria is associated or helps to prevent or protects the intestine from damage caused by infections. So we can see that upon the introduction of the strain, the levels of lactobacillus bacteria dropped from 14% to 4%, meaning that the intestines are more exposed or more prone to getting damaged by any infections that might arise in the body. However, ciprofloxacin was able to elevate the levels up to 11%. Manihot esculenta was able to raise the levels marginally to 5, and the combination was able to also raise the levels marginally up to 6% in the body. Conclude, Manihot esculenta, just like ciprofloxacin, has increased the levels of the good bacteria such as bacteroidal, lichenospiral, and lactobacillus, while suppressing the levels of the bad bacteria such as oscillospiral. Therefore, we can safely conclude that Manihot esculenta has a high potential to be used as an alternative form of treatment to Uropathogenic Escherichia coli and probably most other UTIs, the same way as ciprofloxacin. And these are the references that I have used in compiling this PowerPoint presentation. And for more information, please feel free to contact me on the above email. Thank you.